The overall goal of this procedure is to demonstrate a flexible method of re-engineering viral surfaces via click chemistry. In the first step of this procedure, human embryonic kidney cells are infected with type 5 adenovirus and incubated in the presence of either the unnatural amino acid azidohomoalanine or the azido sugar paracetylated N-azidocetylgalactosamine. After 2 days, the azide-labeled adenovirus particles are harvested and purified. Next, the chemospecific azide alkyne cycloaddition is used to ligate fluorescent probes to the surface of these particles. As explained in the written protocol, this is performed using one of two possible ligation methods: strain-promoted or copper-1 catalyzed azide alkyne cycloaddition. In therapeutic applications, these attachments can include polypeptides, polymers, or small molecules. In this experiment, a fluorescent probe is used so that ligation of the probe onto capsid proteins can be demonstrated via SDS page.
There is significant interest in developing viruses as gene delivery agents, particularly in the context of therapeutics. Gene delivery, oncolytic therapy, vaccine technology. In that context, tuning the virus-host interface is particularly important in terms of getting the appropriate properties for that therapeutic. Tissue targeting, immune stimulation, a number of other factors are important in terms of tuning the host-virus interface. In that context, we've developed a two-step labeling protocol. The first step is the metabolic introduction of unnatural amino acid or unnatural sugar. This installs an anchor point that we can then further elaborate with very, very highly selective chemistries that we can then append those viruses with essentially any functionality we'd like to without significantly perturbing the virus infectivity or overall fitness, which is a major bar.